So as many of you know, we're broadcasting live from the Peabody Museum, which is still under renovation, but will be opening later in the spring. But regardless, we're really excited to be able to even though the building's under renovation, the research and activities are still ongoing inside the museum. So we're bringing you live content from within that context. A quick intro, I'm Katrina Damrell. I'm a grad student at the Yale School of the Environment, and I'm also the Research Spotlight Assistant at the Yale Peabody Museum. And the Research Spotlight series is, as I mentioned, an opportunity for the museum to highlight the many research projects underway using the Peabody's collection. And so before we get started, I just want to do a few housekeeping items. Today, we're using Zoom webinar, so the chat function is disabled, but if you have a question, please use the Q&A function. And David, who's off screen here, he will be monitoring the Q&A, so you guys will be tracked, and we'll have time at the end of the talk with Dalton to answer those. I will say we often have too many questions and not enough time. So if we don't get to your question, I apologize in advance. Also, our program is being live captioned. So thank you, Michelle, out there for captioning for us. And if you want to turn the captioning on, there should be a button on the bottom of your Zoom window towards the right. So that's also another option. And with that, I'd like to introduce our speaker today. We have Dalton Meyer. Dalton's a PhD candidate at Yale's Department of Earth and Planetary Sciences and originally from Loveland, Colorado. Dalton's work focuses on understanding lizard evolution, and his research combines evolutionary theory, 3D imaging, and paleontology to study how the most diverse group of land-living vertebrates alive today, lizards, still have unknown origins due to a poor fossil record. So his talk today will show us how 3D imaging has brought new life to two fossil lizards from the American West and filled a gap in our understanding of how of these two distantly related lizard groups. Each fossil hails from a different era, one from 150 million years ago, the other from 30 million years ago. And they each tell us what these early lizards would have looked like, as well as how their modern relatives today have spread throughout the world. So we got to see it yesterday, and I'm really excited to be able to have it here. With that, I'll pass this off to you, Bill.
Thank you very much. I'm really excited to be here today. Let me just make sure that everyone can see the presentation. All right, looks like people can. Let me just adjust something here. Okay, great. So yeah, thank you very much. I'm really excited to be here sharing some ongoing research projects of mine and a project that was actually just published yesterday. What I'm going to be talking to you all about today are lizards, as the title suggests. And they're one of my favorite groups of living animals, and I hope that by the end of the talk, they'll be yours as well. And why am I interested in lizards? I always like to put it into context as to why I think lizards are an important thing to study. And it's because lizards, and I'm including snakes here because snakes are just highly modified lizards, are an incredibly diverse group. They're distributed across the globe and have achieved a huge variety of appearances and lifestyles. So there are over 11,000 living species of lizards, and that makes them the most speciose group of what we call tetrapods, or four-limbed animals, that are alive today. And to put that into some context, there are around 5,500 species of mammals that are alive today, and around 10,000 species of birds. So we're kind of neck and neck with the ornithologists. Right now we're on top, so go lizards. And over the course of their evolutionary history, they've achieved tons of extremes. So one thing that comes immediately to mind are extremes in size. On the left here, we have a living microchameleon, Frechesia, about the size of a match head, one of the smallest egg-laying animals alive on Earth. And in the prehistoric past, lizards achieved size pushing 50 feet plus in length in the marine mosasaurs. They've also achieved a huge range of diets. Now, most lizards are insectivorous, so they're wandering around eating bugs. But within each group, there are many explorations into other dietary avenues. eating hard-shelled prey like our friend in the upper right there. Varanids and snakes have both independently gone into eating large game, and many groups of lizards have also independently gone into herbivory. And beyond their diet, they also have a huge range of lifestyles and habitat use strategies. So a lot of lizards are just terrestrial, walking around, but there are tons of lizards that are arboreal, that live in trees. We have lizards that burrow, they live in leaf litter and kind of crawl around the forest floor. And there are even specialized lizards and snakes that glide from tree to tree. And of course, you can forget the marine iguana, which is adapted to eat algae off of rocks and just kind of grab onto these submerged surfaces. And all of this is to say that lizards, with their beautiful diversity and disparity in form and function, make an excellent natural laboratory for investigating evolutionary phenomena. However, there is an outstanding issue to that. And that is the fact that when we construct family trees of lizards from different data sources, we get pretty different results. So you're going to see over the course of this talk some diagrams that look like this. It's called a cladogram, and you can think of it as a family tree of lizards. And if we generate these family trees with morphological data, that is to say, data that comes from the anatomy of lizards, looking at their bones, their scales, the color of the scales, things like that, we get one result. And if we use molecular data, things like DNA and proteins, we come up with a different tree. Of particular note, in difference between them is the position of iguanians. So if you use anatomy, iguanians come out at the very base of the tree. They're the first emerging lizard groups from that last common ancestor. But if you use molecular data, they come up very highly nested as closely related to monarch lizards. And so these outstanding questions kind of impede our ability to use this system to investigate a lot of evolutionary phenomena. And now this is not a problem that one person can solve, and we're slowly building consilience between data sets. But what I'm particularly interested in is looking at the origins of these major groups, looking at fossils that sample when these things first emerged to get a sense of what the anatomy of the very earliest members of these lineages look like, and how that can inform us as to what the structure of this tree is, and to what patterns lizards have experienced, over the course of their evolution. And so to look at these origins, we have to go back in time and we have to look at the fossil record. And I'm going to start by looking at one particular group, and that is the Anguidae or the Anguioidea, kind of depending on which flavor of the name you like. This group consists of several kind of subfamilies. So these groups include the Diploglossidae. These are the gollywasps, and they're native to the Caribbean, Central America, and South America. We have the Geranotines, the alligator lizards, and they're native to Central and North America. We've got one oddball, the Limbless Aniella, which is the California legless lizard. So as its name implies, it lives in California. That's all we find in today. And then we have the Anguinae, the glass lizards, and these are another limbless clade, and they're found distributed across the Northern Hemisphere, North America, Europe, Asia, North Africa. And what's important to note, and we'll come back to this later, is that the North American and Asian groups of anguines, of glass lizards, are each other's closest living relatives. So within the glass lizard group, the North American and Asian forms share a close ancestry. To look at the origin of anguines, we need to go back in time. And specifically, we're going to be looking at the Oligocene. This is around 30 million years ago. And we're looking at a rock unit that's part of what's called the White River Badlands. And this is an extent of Oligocene and a little bit older rocks that crop out across a lot of Western North America. So you see them in places like South Dakota, Nebraska, Colorado, and Wyoming. And this particular fossil we'll be talking about does come from those Wyoming outcrops. We're looking at the Brule Formation. And so the Brule Formation preserves a glimpse into a changing North America. It's a really interesting time. The warm and wet climate that had persisted since the end of the age of dinosaurs is starting to cool, and even more importantly, it's starting to dry out. And you're seeing the loss of major forest cover in a lot of the middle of the North American continent, and the origin of the huge grasslands that we think of now as part of the Great Plains. And the fossils from these emerging grasslands show a suite of animals. These include early mammals, things like these early horses you see here called Mesohippus, early relatives of cats that are called rabbits, as well as an assembly of reptiles, a lot of which seem pretty modern. So you can see in this diagram here, this art, there's a turtle in the lower right. But among those more familiar reptiles, we also have lizards. And one lizard in particular that we're going to be talking about is Parophosaurus poniensis. Now, this species has been described on a number of partial skulls, as well as one specimen that had some of what we refer to as postcranial anatomy. So that's the skeleton that is behind the head. And from those descriptions, we can see that it's kind of defined as it's an anguid lizard. It lacks striations on its teeth, so its enamel on its teeth is smooth. And it was described initially as potentially being limb reduced, although that is again based only on this partial specimen. There have been a couple hypotheses as to where it falls on the anguid family tree. When it was originally described, it was hypothesized to be related to the glass lizards, but in particular, particularly closely related to the North American glass lizards, to the exclusion of the other glass lizards. And what this would potentially imply is that this group may have experienced multiple independent losses of limbs to get this kind of snake-like body form. But in 2008, an alternative hypothesis was proposed that it's actually a close relative of the alligator lizards and is on the stem of the Gyrodontian group. Now, I've had the distinct pleasure to work on a new specimen that is at the Yale Peabody Museum, which represents the first complete skeleton of this taxon. And by doing this, I'm allowed to look at anatomy that had been previously unseen and really probe where this falls on the family tree. So why do we say that this is Peabody? Well, a number of features lead us down that line, but most importantly, if you take a close look at its teeth, you can see it has smooth enamel, there's no striations. But looking at the full skeleton, we can say that it actually really isn't limb reduced. It has a nice kind of full complement of proportionate limbs. But how do I investigate these fossils? I mean, obviously looking at them and taking detailed notes and looking at the anatomy is great, but there's a lot of that that's obscure. And so I use a technique that's called micro CT scanning. And this is, if you've ever had a CAT scan, the same principle. It's using X-rays to make a 3D model. And now instead of a medical CAT scan where the patient lies still and an X-ray source rotates around them, in this case, the X-ray source is fixed and the sample is put on a rotating stage. And you take a number of photographs with the X-ray to generate these slices that you can then digitally reincorporate into a 3D model. So to reorient ourselves, here's the specimen again. And this is what it looks like after CT scanning when it's assembled with the computer. So you can see really nice high resolution anatomy, but we can do even better with the scans. So once it's digitized, you can remove the surrounding rock and get a glimpse of the skeleton that is obscured by matrix that you could otherwise not prepare away carefully or safely. We're still obscured though, because anguids have what are called osteoderms. They have these bony plates that are in the scales that you can see covering the body of this lizard and hiding a lot of that internal anatomy. And with the CT data, we can also remove those and get a glimpse just at the skeleton. And what I can do with this data is then go in and digitally segment out and isolate individual elements of the anatomy for close study detail. We can look at surfaces that are otherwise obscured. We can look at the interior of structures like the brain case and the jaw, and a host of new data that we can use to incorporate this into family trees of lizards. And that's exactly what we did. So where does this fall on the tree? We've ran this in two different data sets, and in both cases, what we're supporting is that Peabody is the sister to all extant anguids, the glass lizards here in green. Not in fact the Gyrodontians, the alligator lizards shown in red. But what's important to note that our data supports it not being sister to just the North American anguids, but actually to all of the living glass lizards. So this isn't an exclusively kind of North American group. This seems to be representative of the ancestral form of all living glass lizards, which if we remember are all legless forms. And so it gives us a glimpse into the early evolution of anguid features. These are things that are appearing in the skull that precede the loss of the limbs. And they include the development of this hole at the front of the snout called the premaxillary foramen. The bone at the very front there is the premaxilla, the tip of the jaw, and then you have the two maxillae on the side called the teeth. This is a pretty esoteric one, but in cross section, you can see this bone, the prefrontal in blue, is cupping behind the nasal capsule. And then if we look at the jaw, this bone that I've labeled here, sphenial, is starting to reduce its extent and kind of decrease its size. And these are all really minute features that aren't particularly useful for identifying an animal, especially if you have one that still has skin on it. But they're showing that this is actually the road that this group is taking before it then loses its limbs sometime later. And we can also learn a bit how anguid lizards got around the world. So this is a map of the world 30 million years ago showing where Peabody lived in North America. And we can infer from this kind of a pathway that they would have had to take to achieve their modern distribution. So from the stock that gave rise to Peabody, some of those lizards must have at some point, either after the emergence of Peabody or possibly before, migrated from North America into Europe. And it's within Europe that we see the emergence of the group that gives rise to all living glass lizards. The European glass lizards obviously stick around, but the ancestor of the group that contains the Asian and North American glass lizards would have at some point migrated into Asia. And from there, the Asian group sticks around, and the origin of the North American group arises in Asia, and then crosses over the Bering Land strip into North America to then give us the distribution of glass lizards that we have today. And none of that we can know about the fossil record, giving us a hint of where these groups are originating. So now we're going to move on from anguids, and we're going to look at another part of the lizard family tree, and this time we're going to be looking specifically at geckos. Now, geckos are a charismatic group. They're pretty cute. A lot of people enjoy and like geckos, myself included. And it's important to study geckos because regardless of which data source you use, they are found near the base of the whole tree of lizards. They're either the second emerging group or the first emerging group. And so the ancestral form of geckos is going to give us a good glimpse into probably what the common ancestor of all lizards looked like. So we look at modern geckos and their distribution. We have the first major group of geckos are the eulephorids. These are things like leopard geckos, and they're found across the northern hemisphere, North America, Central America, North Africa, and Asia. There is the next major group. This is called the pygopodoids. This includes the wingless geckos, and this group is found only in Australia and surrounding Australasian islands. There are these two groups, the spherodactylids and the philodactylids, and they're pretty cosmopolitanly distributed in the Americas, North Africa, Europe, and Asia. And then there are the geckonids, which are probably the most familiar geckos for a lot of people. And I haven't put any lines on here because it would just get too messy. They're found all over the world. And to investigate the origin of geckos, we have to go further back in time, this time to the Jurassic. And we're looking at around 150 million years ago now. And we're looking at rocks that come from the Morrison Formation. This is a famous Jurassic formation that covers a huge swath of Western North America. And we're zooming in particular at Dinosaur National Monument that sits at the border between Utah and Colorado. This is a photo of the famous Dinosaur National Monument quarry from the outside, showing the surrounding landscape and the rock units. And if we go inside that building, we can see the famous Dinosaur National Monument quarry wall. Now, this wall contains the bones of hundreds of in-place dinosaurs. If you ever have a chance to go there, I highly recommend it. It's an incredible place. And if we take a look at what's living in the Morrison Formation, we have a suite of pretty well-known dinosaurs. These include Allosaurus, the big predator of the Jurassic, sauropods like Apatosaurus there in the back, the really common Camarasaurus over there on the right, and Yale's very own Brontosaurus right up here front and center. There's also other famous dinosaurs like Stegosaurus and a host of non-dinosaurian taxa as well. These include flying pterosaurs, crocodilomorphs that look a lot like modern crocodiles, and then also, of course, lizards. So there are two skulls that have been found at Dinosaur National Monument that represent fossil lizards. And these are Dino 14864 on the left and Dino 15914 on the right. Those are their specimen numbers. Now, both of these specimens, when they were originally described, were referred to the genus Paramasilotus. This is a genus of fossil lizard that was first found in Europe, I believe in England. And the describers of these said, hey, these look pretty similar. We think that's what they are. Me and my colleagues decided we wanted to take another look at these using modern techniques and see if we could get any more anatomy out of them. So once again, we turned to the CT scans. And Dino 1486, or 14864, excuse me, that skull that was on the left on the prior slide, we described last year and named Eoscincus ornatus in a paper led by my colleague Chase Brownstein. And then it was time to turn to Dino 15914, the other skull. And so this is that drawing from the paper on the left, and this is the real fossil on the right. And to give you a sense of scale, here is the lizard, and here is the Dalton. It's a very small fossil. They have to use a special magnifier in the exhibit to let people see it. And once again, we turn to CT data to give us access to unknown anatomy. And so this is the fossil CT scan in position as it's in the rock. And what we're able to do is reassemble that to a semblance of what this lizard probably would have looked like, a more lifelike position. We've named this new species Helioscopus dickersoni in a paper that came out just yesterday. Very excited about it. The name means Dickerson's sun gazer, and that comes from this feature that's in the circle here. That is the pineal foramen, and it houses what we refer to as the parietal eye. Now, this is a light-sensing organ. It has a lot of the anatomy of a regular eyeball, but it doesn't have an iris to contract, so it doesn't form images, but it senses light, And lizards use this to regulate their physiological processes that are dependent on light. Things like thermoregulation, am I too hot, am I too cold, do I need to go in the sun? And also to track day length and regulate seasonal physiologic processes. Like, when is it time to breed? When is it time to hibernate if I'm going to do that? And so from this, we can infer at least that Helioscopus was probably living during the day and depending on the sun for these processes. And it's Dickerson's sun gazer, named after both Mary Cynthia Dickerson, who was the first curator of herpetology at the American Museum, as well as my late grandmother and great aunt, Shirley and Helen Dickerson. And so we did the same thing. We did Paropsaurus. We put it into a family tree. We took all this new data. And what do we find? Well, we find that it's at the base of geckos. And this is if we look at just its anatomy, this is if we constrain the living organisms to what DNA is telling us. And it's also true if we use a more complex computational method that accounts for the age of the fossils in the analysis of its position. In all cases, Helioscopus is closely related, extremely closely related to some German fossil lizards that come from the same locality that produces Archaeopteryx. And they are found at the very base of what we call the total group of geckos. So all lizards that are more closely related to geckos than to anything else. And if we take a closer look at this family tree, there's Helioscopus right there, we can learn a little bit about the assembly of the gecko skull. So geckos have pretty modified skulls relative to other lizards. And we can start to see the acquisition of some of those features. I'm just going to highlight a few here. Starting with that penile foramen, modern geckos have closed that up. And we think that that's probably because most living geckos are nocturnal. So they're no longer relying on the sun to regulate all of these life processes. And they close that hole. It's still there in Helioscopus, but we infer that there's probably one loss of that just outside of what we call the crown gecko group of all living geckos. Other features, another kind of esoteric one here, is the margin of the prefrontal. In modern geckos, it extends underneath the brain. In Helioscopus, it's just the inner margin is a straight line, that ONM that's figured there. And what we see as we track that feature along the tree is that it goes from a vertical margin, slowly sloping in, sloping in, sloping in, and then finally achieving its position in modern geckos. And we call that a stepwise acquisition. You're taking steps towards getting this feature. And that's kind of classically how we think evolution works, and a lot of features in this tree behave that way. But some other trees don't behave, or some other traits don't behave so nicely. If we look at the roof of the mouth of Helioscopus in a modern gecko, you can see in both cases that there are no teeth on the roof of the mouth. That might sound weird because we don't have any teeth there, but a lot of reptiles and a lot of lizards do have teeth on the roof of their mouth. Geckos do not. Helioscopus does not, but other fossil taxa that are more closely related to modern geckos still do have those teeth. And so we see this feature being lost multiple times along the way to the modern group of geckos before finally being kind of locked into place later up the tree. And we refer to this whole pattern that we're seeing with all of these features as more mosaic evolution. Some things are appearing, some are disappearing. It's more complex than you might assume without the fossil record. And we can also learn about the origin of geckos. So turning to the distribution of geckos in the world, our data infers a European origin for the total group of geckos, and Helioscopus is the first known incursion of this group into North America. It predates the earliest other gecko fossils by about 100 million years. And this is something we would never have known if we hadn't found this fossil and analyzed it. We also infer from this an Asian origin for all living geckos, that crown group of all modern geckos. That common ancestor probably lived in Asia, and that provides two scenarios for the dispersal of this weird Australian group, the Pygopedoids. We infer that this group originated in the kind of mid-Cretaceous, and at that time, Australia was still connected to Antarctica. And this gives us two routes for these lizards to take in order to get to Australia. One would be oversea from what will become Southeast Asia, and the other, which is probably a bit more easy for these small animals to make, is actually walking overland through Africa, and then crossing into Antarctica, and then crossing from Antarctica into Australia when those continents are still connected. And then they essentially became trapped in Australia as it broke off and moved northwards. And so I hope that you now have a better appreciation of the diversity of lizards, their complex and fascinating evolutionary history, and that you have a new sense of appreciation for them, just as I do. I've been privileged to talk to you all today. Thank you so much for attending. To thank all of my supporters, from collection support, co-authors, my colleagues at the museum and university, and again, thank you all for attending.
Awesome. Woohoo! go dolphin. That's so cool. You want to take a seat next to me? We can answer some of the audience's questions.
Absolutely.
And as you guys might be able to tell, the sun is setting here in New Haven, so sorry if the lighting's a little dark over here, but
I'm gonna put this
we're
on
good.
screen share.
Yeah, perfect.
There we go.
Yeah, thank you. It's salted.
Mhm.
Awesome, cool. So let's see what we have for some questions here. That was such an informative talk, first of all, and thank you so much for all the different imagery. I thought that was such
Oh,
an
thank you.
engaging presentation. Let's see, actually, speaking of the emergency services, speaking of the slides and the content, Michael is asking about the Areocealus, I'm sorry, I'm not
No worries.
as pictured on the Age of the Reptiles mural. And so maybe that if you feel like pulling up your slides, feel free, but.
Yeah, so I don't think I have any slides that, that would help with Areocealus,
Okay,
unfortunately.
Areocealus.
But yeah, that Areocealus is an animal from before anything that I studied, frankly. So lizards, we think, originated sometime in either the late Triassic, mid-late Triassic, maybe earliest Jurassic, depending on which group of lizards you're really interested in. Areocealus is an animal that comes from before that. Areocealus predates what we call the Mesozoic, or the age of dinosaurs. And it predates my knowledge a little bit too. Areocealus, I believe, is an early reptile of some kind, but my colleague Kelsey Jenkins is actually working, and I'm working with her on a project that is redescribing the tree of all reptiles, and especially very early ones. So there will be some insights into Areocealus that will hopefully come out soon.
That's so exciting.
That I unfortunately can't share those, but as it's depicted in the Age of Reptiles mural, it kind of is like a creeping lizardy salamander thing, and it probably looked more or less like that.
Did it also have a third eye?
I believe so.
How did you describe that? Helioscopus was the.
Yeah, so the pineal eye, or the parietal eye, they're kind of used interchangeably. I'm pretty sure Areocealus had one of those. So that's something that's ancestral to not just reptiles, but pretty much all tetrapods. So even early members of the mammal line also have that eye, and it closes up in about the Triassic.
Okay. Interesting. Okay. So another question here is, was the shared ancestry of the anguines from Asia and North America determined based on morphological or molecular analysis, or a combination of both methods?
So it's been a combination of both, and both independently. So it's something that we can detect using morphology. It's even pretty more strongly supported with molecular data that shows that, yeah, that the Asian clade and North American clade are each other's closest relatives. And if you take both data sources into account, you still find that.
Does that kind of double confirmation in different methodologies? Is that a pretty common strategy these days?
It is, yeah. So more and more people are trying to use what they call combined data, which is combining genetic information and morphological information to produce a tree. Because we think that genetic information is obviously an amazing and powerful tool, especially for looking at living organisms. But fossils represent 99%, well, the sample that we have, right, of the 99% of all life that has gone extinct. And the only way to incorporate fossil animals into our trees is to use morphological data. And so those combined analyses are a powerful tool that let us use kind of the best of both worlds to investigate these kinds of questions.
We have a question from Sam about what's the deal with diamids? I'm sorry, guys.
No worries.
I'm really butchering these. Again, School of Environment students. I've never heard of them and didn't realize they might be the earlier diverging lizard.
Yeah, so I will go through my slides here.
Yeah, please.
To pull up that picture, just to give everyone a glimpse. I don't talk about diamids much in my slides, and I don't work on them myself particularly closely. But diamids are what we call worm lizards, and when I show the picture, you will see why. So I'll pull up this tree. Let me just get the screen share option on. So yeah, they are in the upper right of the tree there. On this particular tree, they're just outside of geckos at the base of the group. They are a limbless group of lizards that are very small. They're not particularly common and abundant, and they're very tricky. The easy answer is we just don't know. So now that everyone's gotten a look at the word lizard, I'm going to turn the screen back off. But the issue with dibamids is that morphologically, they don't tremendously closely resemble any other lizards. They're so highly modified for their burrowing lifestyle and by becoming so miniaturized that their skeleton is extremely strange. And so, you know, when people started doing molecular phylogenetics on lizards, they're very hopeful, oh, we'll figure out where to go. And the answer is we still haven't. So we're confident that they're near the base of the tree, but some molecular results find them as the very first group of squamates. Some results find them as the sister to geckos. So they find a clade of geckos and dibamids being the oldest. Sometimes geckos come out and then dibamids come out. And it's an ongoing area of research from basically everyone who's interested in squamates is trying to figure out where they go on the tree, because we just don't know, except to say probably pretty early.
Wow. So cool. Let's see, David is asking about if the Cretaceous gecko fossil had a paired frontal bone, or did that break off the fossil? Did the Cretaceous gecko fossil have a paired frontal bone, or is that a breakage on the fossil? Okay, so I guess if we're discussing Helioscopus in this fossil, that one is paired, it's not broken. There are cracks along it, definitely, but there are margins that you can see in the CT data that are distinct and clear. There are some Cretaceous fossils. I'm not sure exactly which one. If the asker is mentioning one of those in particular, there are Cretaceous geckos that have a fused frontal. So that's something that we see in modern geckos. That bone in the midline skull fuses up into one. That happens in a ton of lizards. Okay.
And then there is, I believe, Gobekko that may have a crack, but to the best of my knowledge, most of the Cretaceous geckos seem to already have fused the frontals, but the Jurassic ones, it's more variable.
Okay, and I have a question about I actually have a question about the sampling and the collection of the fossils. You mentioned both of the fossil samples that you guys have are quite rare. How did the sample get collected? Was that part of your field work, or that's
a good question. They both predate me.
Okay.
The Perovisaurus fossil was acquired by the Peabody Museum actually right as I was starting my PhD.
Okay.
And so that came from some collectors in Wyoming who the Peabody acquired that specimen from. And then the specimens at Dinosaur National Monument were collected back in the late 80s or early 90s. I'm not sure exactly when they were collected, but certainly they were described in the 1990s. And collecting small fossils is a bit different than collecting something like a dinosaur. So the kind of classical image and thought of a paleontologist is going out to some barren area and wandering around until you see something come out of the ground. And that is how it happens for the most part. But that's much more easy to do if the thing coming out of the ground is like this big, instead of a tiny little lizard. So what often happens is you'll find a larger fossil and then start to explore that area in more detail, kind of on your hands and knees. And maybe if you're really, really, really insanely lucky, you'll find something articulated like Perovisaurus. More often, you're going to find isolated bits, or more fragmentary bits like Helioscopus. And even more common for dealing with lizard fossils is what we call screen washing. And that's where you collect, you're not really collecting fossils per se, I mean you are, but you're basically taking samples of sediment and then sieving them through screens of different size until you get a sample that's size sorted. You can look at it under a microscope and then pick out from there what's bone, what's rock, and then you can identify what those are. So with really small stuff, that's probably the most common method right now that people use. Okay,
and when you're sorting that way, is it, if you find bone fragments, is that kind of like the green light to literally go back to that source and see if you can find more of a cohesive fossil?
Yeah, it can be, and that's becoming more common even with CT data. The lights have dimmed. I apologize.
Yeah. The room has its own schedule.
What's becoming more common practice is both returning to sites to collect more sediment to screen wash, but also if you're finding isolated bits in your washed samples, to go back and try to collect more kind of cohesive blocks, and then taking those blocks to some kind of scanner, either like a CT scanner or a synchrotron, to 3D image the entire block. Because the one downfall of doing that kind of size sorting is that anything is associated. If you have like a skeleton that's got a head and all this little backbones and some arms, once you break apart the rock, that's all going to fall out of association. So it's becoming more common now to just take chunks and see a little bit poking out, and then let's scan this before we do anything else and see what we have.
Okay, cool, thank you, that's a helpful visual. Tim is asking about, I guess, kind of similar to what I was just talking about in terms of the field work. It seems like there's a lot of lizards coming out of the Morrison Formation recently. Do you think any more gecko and geckotens might be in store?
Yeah, geckos. You got it. Possibly. And there's a lot of lizard fossils coming out of the Morrison, and it's a really exciting time for that. Some of them I'm working on, some of them I'm not. Unfortunately, well, I can share the one that I'm working on right now. At this point, we don't really know what it is. And I can't speak for what other people are working on, I don't know. But I would suspect, given the fact that we have one gecko now, and from, as I mentioned, this is closely related to the German fossils from what's called the Solnhofen archipelago. That's where Archaeopteryx is from. There's a ton of lizards from there. A lot of them we find as geckos. So it seems that this is a pretty diverse group early on, and I would not be surprised at all if we find more and more.
So exciting. Okay, Eric is wondering why geckos are so universally well adapted. Why they're found so universally.
That's a good question, and there's probably a lot of factors to that. So they're a very old lineage, so they've had a chance to disperse around the world, as we discussed. That helps. They have, on occasion, achieved some very bizarre adaptations. So the limbless ones, for example, in Australia, they're clearly doing a different job and a different thing than any other gecko. And so they've exploited a resource that other lizards simply haven't. Another big advantage for a lot of geckos, though, is that they're small and they're nocturnal. And not a tremendous amount of lizards are nocturnal. So they're exploiting the available resources at night that other animal groups maybe aren't. And so, you know, they're in the same kind of competition with mammals and other reptiles that all lizards face, but they're doing their own thing with respect to other lizards. And the other thing about geckos is many groups of them have independently acquired the really specialized toe pads. So if you look at climbing geckos, you see their kind of really bulbous feet. They have these specialized scales that have all these tiny microscopic projections that let them cling to pretty much any surface, even really smooth surfaces like glass. And that lets them exploit vertical environments really well. They're great at climbing trees. And also, as urban environments kind of ingress into previously non-urban environments, they're pretty well adapted to interfacing with the urban world as well, because they can climb man-made structures and do more or less similar things that they would be doing in the wild.
Kind of a generalist approach. That's really cool. And the diurnal adaptations seem smart too. Okay, and then David is wondering, what does Periosaurus tell us about the timing of the leg loss of penguins?
Yeah, so that's an interesting question, and there's a couple things we can interpret from that. So Periosaurus is from the Oligocene, right? 30 million years old, and we have then some good reason to expect that perhaps the loss of limbs in that group occurred after that, so sometime within the last 30 million years. There is remaining question about that, though, and it's important to consider that the fossil record is imperfect. And what we don't yet know is if Periosaurus represents kind of a North American clade that then had sisters that went over into Europe and lost their limbs, if that group lost its limbs in North America and then went into Europe, or if the total group actually had this split before Periosaurus occurred, and Periosaurus is just a remnant of that later group. So Periosaurus may have split off from the line leading to Periosaurus could have split off maybe 40 million years ago or 50. And we're just sampling where that group went, and then that other group may have gone and lost its limb at some other point. So in terms of the timing of limb loss, we have some idea that maybe it's within the last 30 million years, but to really confirm that, we need some better samples of even closer relatives and then from Europe. And we have fossils of those, but right now we really only have skull material and no bodies, so
Yeah.
it's hard to say.
Okay. Okay, let's see, we have time for maybe one more question. Let's see. Well, we have a question from Dottie, asking about whether dinosaurs are lizards.
That's an excellent question, because the name dinosaur means terrible lizard.
I did not.
Yeah, but that's only because the root saurus, meaning lizard, is kind of treated more broadly to mean reptile, because when ancient people were looking at reptiles, there wasn't a great understanding of that there are actually multiple groups within it. It was kind of, oh, here are all these creepy, scaly things, and we don't like them very much, so they're all lizards. And so to answer your question, Dottie, dinosaurs are not lizards. Dinosaurs are what we call archosaurs. So there are two main groups of living reptiles. Turtles are a bit of a mystery, so we're going to not talk about turtles.
Okay.
They are reptiles.
Okay.
But there is one line which we call lepidosaurs, and that contains lizards and snakes, as well as the tuatara from New Zealand. And then on the other side of reptiles, we have the archosaurs. And the archosaurs today include crocodilians and birds. And dinosaurs are on the bird side of that archosaur lineage. So dinosaurs are reptiles, but they're not lizards.
Okay, well, I just learned something new too, since my childhood, I did not realize that. Well, thank you so much, Dalton, and I think that comes to a wrap for us timing wise. I see there's a couple more questions here, but if you guys want to email Dalton directly, feel free.
Absolutely.
Thank you for that. And if you enjoyed this talk today, I also want to note that we will be updating our Yale Peabody YouTube channel with a recording from today. And so that'll be up in the next couple of days. And then also you mentioned your colleague Kelsey Jenkins.
Yes.
She is a colleague of Dalton's, and she also was featured in the Research Spotlight series last semester. So if you want to learn more about the what's third eye?
The third eye, the parietal eye.
The parietal eye, there we go. The parietal eye, feel free to check out our YouTube channel because that's up there. So it's really, really interesting. And the Research Spotlight series continues next week. We have Mansa Shrivastava. She's going to be talking about her research in the Himalayan mountain range, and she's from the Department of Ecology and Evolutionary Biology, and that will be around alpine plants. So again, that's next Thursday on December 7th. The email went out today to register, so check your emails, and if you don't see it, you can check our website. And then please follow us on social media, as well as sign up for our mailing list to stay in touch. And yeah, that brings us to the end. And thank you again, Dalton, so much for joining us. It's really exciting to see all the research that you're doing. Congratulations on your latest publication.
Thank you very much. This was a lot of fun. Thank you for having me.
Of course. Bye, guys.